Hello, everyone. This is the Zoom recording for the week of October 26. So in this Zoom recording, I have included blood and the endocrine system. So do not expect any Zoom recording on Thursday because I'm doing both blood and endocrine system today. So for the blood PowerPoints, I wrote down my own, I prepared my own PowerPoint, so you can use this one as your way of preparing for the next quiz. And actually this one is, instead of 37 slides, I make only like 2 or 3 slides for blood. So for these PowerPoint slides, the things I want you to know are first, I want you to know about the components of blood. Blood has two components, the fluid portion and the solid portion. The fluid portion of blood is called plasma. And for the lab, we are more concerned about the solid portion because we can actually see it under the microscope. So the solid portion of the blood is called hematocrit. So within the hematocrit, we find different kinds of blood cells that we can find in blood. And those are the red blood cells, the white blood cells, and the platelets. And each of them has their own names. For example, red blood cells are called erythrocytes, and the white blood cells are called leukocytes. And as you might probably know, leuko means white, cytes are cells, so white cells. The same thing for erythrocytes. Erythro means red and cytes are cells. Thrombocytes are platelets. They are not blood cells by themselves, but they are grouped under blood cells. They are just fragments of cells instead of a full-fledged cell. Thrombo means this is related to thrombin, and thrombin is like the biggest component in blood clotting. So for erythrocytes, I want you to determine the size of erythrocytes compared to platelets and the white blood cells. And as you know, white blood cells are much larger than the red blood cells, but much bigger than the platelets. In terms of shape, erythrocytes, they look biconcave, and their function is to transport oxygen and with the help of hemoglobin. And the other thing I want you to know is erythrocytes do not have the major organelles or cellular organelles we know. For example, they don't have like nucleus, they don't have mitochondria, they don't have the major organelles we know in regular cells. Leukocytes are the white blood cells. On your quiz, I'm sure I'm going to include some of the pictures of white blood cells, and you should be familiar with them. If I give you a microscopic slide of, for example, one white blood cell, let's say neutrophil, you should be able to identify. Saying that, you have to understand how leukocytes are classified. We have two major categories of leukocytes, the granulocytes and agranulocytes. The granulocytes are all the white blood cells whose name includes in fields, for example, neutrophils, basophils, eosinophils, they are fields. The agranulocytes, as you can see, it says A, so this is the opposite. So agranulocytes is those white blood cells that do not have granules. So they induce cytes, for example, lymphocytes or monocytes. For the functions of leukocytes, I want you to take a look at slides 3 and 4 in your PowerPoint slide that I have posted on Blackboard already. So you should be able to know what the function of, say, neutrophil or eosinophil or basophil is, or what the functions of leukocytes, lymphocytes and monocytes are. And related to that, you have to be able to identify what factors might cause certain types of white blood cells to be elevated or to be depressed. That means when do the numbers of certain, let's say, lymphocytes, when does its number increase? Or when does their numbers decrease? So you should be able to know or identify these factors. The other thing I want you to understand is what antigens and antibodies are. Antigens are surface proteins that are found on red blood cells. Let's say red blood cell is like this circular structure, and antibodies are surface proteins. So surface proteins, you are going to find them on top. So let's say one surface protein might be this one. So these are antigens. We find them on top of the red blood cells. So for example, this surface protein might be antigen A, it could be antigen B, or if we are talking about people with blood type O, there might not be anything like this in the first place. And other red blood cells, they might have even Rh factor, a different one. Let's say that Rh factor could be like this one. So antigens, or with their other name, agglutinogen, they are red blood cell surface proteins. And coupled with the antibodies, they help us to determine the blood type of a person. Antibodies, we don't find them on the surface of the red blood cells. Rather, let's say this is the bloodstream. So the blood is going this way. And here's some red blood cell. And here's some antigen on the surface of the blood cell. Antibodies, you just find them in the plasma or in the fluid portion of the blood, just flowing along with the rest of the blood cells, the blood, yes. So you don't find them attached to the red blood cells. They are just moving around within the bloodstream. And they are actually really important in determining blood type too. So regarding antigen-antibody relationship, if you have blood type A, that means you have a surface protein on the red blood cell, which is type A. So you will have antigen A. And in the plasma, you are going to have anti-B, antibody B or anti-B. And if you have blood type B, you are going to have antigen B on the red blood cells. And in the plasma or the liquid portion, you are going to have antibody A. If you have type A, type AB blood, that means you are going to have a red blood cell, and on the red blood cell you have type A, and at the same time you have type B antigens on the surface. So in this case, you have two antigens on the surface of the red blood cell. Since you have both antigens, you will not find any kind of antibody in the blood. So what we are saying is, if you have type AB, you have antigens A and B on the surface of the red blood cells, but in the plasma, there will not be any kind of antibody. If you have type O blood, you will not have these structures. It will be like a completely shaved red blood cell. It doesn't have any, when I say shaved, I'm talking about just these particular surface proteins. So if it is type O blood, you will not have any surface protein. But since you don't have any surface proteins to protect the red blood cell from clumping or from being clotted, what the body does is it will produce anti-A and anti-B antibodies. So the plasma will be filled with a lot of antibodies. The other is the rhesus factor, which is identified in a rhesus monkey for the first time. So there are people who have this particular antigen. It's like antigen A or antigen B. So if you have this Rh factor, let's say this is the red blood cell, and the Rh factor might be some rectangular structure on the surface of the red blood cell. And some people has it, other people don't have it. So this still determines your blood type. So this is what you see when you say negative or positive. We're talking about if you have Rh with, associated with the red blood cell. Let's say if you have this red blood cell and if you have some antigen A over here, and at the same time, if you have Rh factor over here, you are said to have A plus blood type. The plus in this case indicates you have the Rh factor. All right. Okay. The other thing I want you to know is what are safe procedures when we are handling blood, because blood is one of the most probably dangerous fluids if we handle it with bare hands, or if we are exposed to it, it might have a lot of contaminating pathogens. So there are safe procedures when we are handling blood in the laboratory. So on the PowerPoint, I want you to at least identify 3 safe procedures. It might be like wearing gloves, putting contaminated lancets in the biohazard material. But just I want you to at least come up with 3 safe procedures, or just be familiar with the safe procedures that are listed in your PowerPoint. The other point that I want you to be familiar with is blood smear preparation. The slides that I'm going to tell you about, like in the coming slides, the microscope slides, they are actually prepared with a meticulous care. So they are not just randomly made. It takes a lot of caution to prepare them. So what I want you to do is just read the procedures, the steps on how to prepare a good blood smear. And when you make blood smear, we usually use right stain. For example, if you remember, when we try to determine if a certain solution has starch, we used iodine to check it. So like that, right stain is what we usually use when we try to make a blood smear. So when you use right stain, there might be over or under staining, and overstaining and understaining is gonna determine how clear the microscopic pictures that you are going to take are going to be. So let's say if you waited too long, probably there might be overstaining, and when you try to see the blood cells under microscope, they might be dark and might be difficult to differentiate. And in the opposite, let's say if you didn't give it time for the right stain to mix with the blood cells, then that means you are going to have understaining because you didn't give it ample amount of time for the stain to mingle with the blood cells. So I want you to determine how long should the right stain should stay in the smear. Usually it is around 2 minutes. So anything less than that is going to lead you to understaining, and anything greater than that is going to lead to overstaining. The other topic is differential white blood cell count. White blood cell count, this is a procedure we follow when we wanted to know the percentage of white blood cells in a certain blood sample. So in your PowerPoint, there is a picture that shows you how counting is done. So for example, if you are under a microscope and when you try to count, the first thing you do is you start from the top and you go down counting the cells. And then you go to the right, and then you go up, and then you go to the right, and then you go down. While you do that, you are counting. So, for example, if I ask you to count at least 50 white blood cells, this is how you are going to do it. This helps you not to count the same cells twice. So you go down. When you are done, you still count. You go this way, and then you are counting up, and then you are counting to the right. So let's say you counted 50 white blood cells. So usually when somebody tells you to count white blood cells, at the same time, they will give you, let's say if you count this many or white blood cells, the expectation is that, let's say, 50% of those white blood cells are going to be neutrophils. The other 20 might be lymphocytes. And you might, and at the same time, they might tell you you might be less likely to find basophils because basophils account only less than 1% of the total white blood cell count. So what I want you to do is I want you to familiarize yourself with the normal ranges of white blood cells. And the other one is hematocrit preparation. As I have said in the first slide, hematocrit is the solid portion of the blood. That means it is directly related to the volume of the blood cells, including white platelets and red blood cells. So hematocrit preparation is based on volume. This is the most important component of hematocrit preparation. It doesn't depend on mass, it doesn't depend on anything else. It depends on the packed volume. Let's say if this is the micropipette used to prepare the hematocrit, and if the hematocrit was, let's say, this much, like usually less than halfway, so let's say up to here where the red blood cells, up to here where the red blood cells, let's say the micropipette goes all the way up to here, and you use a sponge to block it. So let's say you measure from here, and this is 100%. So, and let's say this one is 45. And in this case, we are not talking about just, it happens to be related to the height, but we are actually talking about the volume. If you can measure the volume of this whole thing, the question is what percentage of this total volume is liquid and what percentage is solid. The solid portion is going to tell you the hematocrit. And usually hematocrit preparation is done through centrifuge. So you will prepare several micropipettes with a sample of blood, and then you centrifuge it, and then you determine the amount of hematocrit. There are normal values that are posted on your PowerPoint on Blackboard, so you should be familiar. And at the same time, there are gender differences. Women tend to have less hematocrit compared to men. The same thing for hemoglobin. Men tend to have higher levels of hemoglobin or hemoglobin content in a red blood cell. And you should be familiar with how hemoglobinometer works. Hemoglobinometer works through color map. For example, let's say you have blue over here on the hemoglobinometer, and then the blood came out like this. So the hemoglobinometer will tell you when you turn on the knob. There is a knob in the hemoglobinometer, and you try to turn it until the two colors become equal or the colors match. Let's say if this green, this one is turning to green, or this second one is turning into blue. It is a color match game. So when the colors match, when the color of the blood and one of the colors that's found in the hemoglobin, when they match, you stop and you see the reading, and that tells you the amount of hemoglobin in that blood sample. Okay, this is all regarding blood. The only two things I want to remind you is in your Blackboard slide, there is something that talks about packed cells. When we say packed cells, we said we have antigens and we have antibodies. We have antigens and antibodies. When we say packed cells from the blood, if you keep the red blood cells with the antigens, but if you can remove the antibodies, that is called packed cells. When you say packed cells, it means blood without any antibody, like inherent antibodies. Let's say pure blood type is A, that means you are going to have anti-B. But for, let's say, by some technology, if you remove the antibodies and then if you keep just the blood with the antigen, then the cell content you gained is called packed cells. The last thing that I want you to know is what are the red flags in blood transfusion. Obviously, if you are type A, you don't take blood from a type B person. Or if you are type B, you don't take blood from a type A person, things like that. Or if you are Rh positive, if you are Rh negative, taking blood from an Rh positive person might be okay for the first time, but it might not be okay for the second time, because the second time, when you try to have a baby for a second time, actually your body has produced an antibody for the Rh positive blood, and that might lead to blood clumping. So you have to familiarize yourself about the red flags. This is probably the last slide in the whole presentation in your Blackboards. Okay. This is all regarding blood, and let's go to the endocrine system. Endocrine glands are, endocrine system contains endocrine glands. These glands are ductless glands. They don't have their own ducts or canals, so their secretions, which are mainly hormones, are sent via blood vessels into the bloodstream. And you should understand how hormones are transported from the secretory cells. Let's say if the hormone is produced in the pituitary, how it goes or how it ends up going to, let's say, the testes or the ovaries. So you have to understand how hormones are transported from secretory cells to the target organs. And you have to know the major glands of our body. Especially you have to know these four: pituitary, thyroid, pancreas, and the parathyroid glands. Pancreas, as you know, it could be endocrine and exocrine. The main focus is the endocrine. But to understand the endocrine portion of pancreas, you should be able to tell which portions of the pancreas or which parts, which cells of the pancreas are exocrine. You should be able to know that. And the other thing I want you to know is, or are the organs that contain endocrine cells. For sure, you should be familiar with the brain because it contains the pituitary. You should be familiar with the thyroid gland. You should be familiar with pancreas, but not just these. There are other organs that contain endocrine cells, including the liver, skin, the kidney, and things like that. The other point that's included in your slides are the classes of chemical messengers. When you say chemical messengers, they could be anything. They could be hormones, they could be enzymes, they could be anything. So these chemical messengers can be divided into three. The first one are the autocrine messengers. Autocrine means these chemical messengers are used for self-consumption. Let's say if the heart produces something and it just uses it for its own purposes and it doesn't send it anywhere else, that's autocrine function. Or let's say stomach produces some kind of digestive enzyme and that enzyme is not going anywhere. Let's say if it functions within the stomach, that's called autocrine function. Paracrine is when the chemical messenger is sent to neighboring tissues. It's like whatever these chemical messengers are doing, it's not used for self-consumption. It is used for a neighbor or tissues that are nearby. Paracrine, by the way, para means nearby. And the last one is endocrine. This one is like a distant relative. Make it distant. This is a distant relative. So for endocrine chemical messengers, which happen to be the hormones, they are usually sent to distant target organs. Let's say if a hormone is produced in the pituitary all the way up in the brain, that hormone is actually sent down to, let's say, the ovaries or the testes or the kidney. So these happen to be distant relatives. They don't go anywhere. It's not just to a distant stranger. It should be pretty specific and it should be targeted. Saying that, you should be able to differentiate between neurotransmitters and hormones because these two are completely different chemical messengers. Neurotransmitters, as the name implies, they are related to the nervous system, neuro. And usually they are sent from one side from one end of the nerve terminal to another, which we call synapses. So their function happens to be pretty local. Their effect is not that global compared to the hormones. And their effects are usually not long-lasting. They have short-term effects. Hormones, on the other hand, they are related to the endocrine system and they have wider and enduring effects. One typical example is, for example, let's say take a neurotransmitter GABA. So this is related to mood. So if somebody injects you with GABA, the effects are actually very quick. But for hormones, let's say if somebody injects you with growth hormone, it takes weeks, months, probably longer, in some cases years, before you see the effects. And the other thing I want you to know is about the types of hormones. We have three types of hormones. We have steroid hormones, we have peptide hormones, or sometimes they are called protein hormones, and then we have the biogenic amines. Steroid hormones, steroids, as you know, are lipid derivatives, so they are lipid soluble. And usually the names of steroid hormones end in ONE or OL, for example, cortisol or testosterone. Usually these are lipid soluble. Peptide hormones are water soluble, and usually they end in INE. I didn't say like always, it says usually. For example, take insulin, or you can think of thyroxine. Even glucagon is included under peptide hormones. Biogenic amines are like the peptide hormones. They are made from proteins. By the way, steroid hormones are made from lipids. Peptide hormones, as the name implies, are made from proteins, and these one are also made from proteins or amino acids. But these are highly modified amino acids. The ones we use for biogenic amines, they use highly modified amino acids and several amines to make these hormones. And most hormones that are classified under biogenic amines have like they are very common in psychology. For example, epinephrine, norepinephrine, these are related to the fight or flight response. Or we know histamine, we have serotonin, even thyroid hormones fall in this category. And they are water soluble like peptide hormones, except thyroid hormone, which is a biogenic amine, but at the same time, it is not water soluble. So these are the types of hormones. And the other one that I want you to be familiar with is the 462 mnemonic. The 462 is, we know we have 4 hormones of the hypothalamus, 6 hormones of the anterior pituitary, and 2 hormones of the posterior pituitary. The 4 hormones of the hypothalamus are the thyroid releasing hormone, and the gonadotropin releasing hormone, and then the corticotropin releasing hormone, and then we have the growth hormone releasing hormone. So the thyroid releasing hormone is related to thyroid stimulating hormone. The gonadotropin releasing hormone is related to the luteinizing hormone and follicle stimulating hormone. The corticotropin releasing hormone is related to the adrenocorticotropic hormone and the prolactin. This one is related to prolactin and adrenocorticotropic hormone. Growth hormone releasing hormone is related to growth hormone. So these are instructions for the pituitary. Like these all are instructions for anterior pituitary. These all are instructions for other organs that are found far away from the brain. For example, for prolactin, its target is the mammary glands or the breasts. Thyroid stimulating hormone is thyroid gland. Luteinizing and follicle stimulating hormone, the target organs are the gonads, that means the testes and the ovaries. Adrenocorticotropic hormone, these are adrenal glands that sit on top of the kidneys. And then the growth hormone is its target organs are the liver, the muscles, bones, the areas where you need growth. And the 2 hormones of the posterior pituitary, these are not actually secreted at the posterior pituitary. They are not produced at the posterior pituitary. Rather, they are produced over here at the hypothalamus, but they are stored and then secreted at the posterior pituitary. So it is made in China, but it is stored and distributed somewhere else. So it is made in the hypothalamus, but actually it is stored and then released from a different location, which happens to be posterior pituitary. I have two, I think they look like announcements. For example, one announcement is the last slide on your PowerPoint has the wrong picture because it talks about parathyroid hormone. But the picture on the slide is a picture of the pancreas. So just remember that. And the last one, I have posted at least 4 or 5 video links, and each of them are microscopic slides. So there is one link that contains microscopic slides of white blood cells. So it shows you how neutrophils look like, basophils look like. So it gives you a time to think about it, and then you can answer. This is like a game. And also there is a video that has a microscopic slide of pituitary, thyroid, and parathyroid glands, as well as the pancreas. So I expect you to go over these slides, these videos, because I'm 100% sure I'm gonna test you on those slides. That's all I have. I will post some other announcements regarding your quiz.